Yeah, hi everyone. Good to see you in person. So I met you guys over Zoom, and today it's gonna be all spatial. So it's gonna be a lot of crash course into spatial genomics and proteomics, how the technology works, a little bit of a glimpse on how we really deal with the data, or if you were to use the spatial omics in your lab or for your projects, like what kind of considerations that you guys want to make. Um, but even before you go in, for those who might need a little more background, we probably have a very familiar knowledge with this fruit analogy, where we started from all the way from bulk learning sequencing, where we almost like had a smoothie where we understood each sample as counts per group. So each sample, which could involve a lot of cells and tissue types and mixture of everything, got blended and measured into how many counts or how many gene expressions that this sample has. And then we had single cell, where we now are able to divide this into a single fruit, or how many apples do we have, how many kiwis do we have, into a different cell types and understand counts per cell. And then we're now talking about spatial transcriptomics or spatial proteomics, where we now not only understand how many things we have in the sample, but also how it's spatially organized and what kind of components and what kind of orders that these cells are, organizations that these cells are having in a given sample. So even going further into the technology itself, it started from very early, when like 1960s, when there was a microscope and like doing a measurement of all different RNAs and single molecule measurements. So this was I would say, the very first spatial omics, where you can measure ribosomal RNAs or messenger RNAs using a radioactive labels, which then become more of the fluorescent labels onto the cell. So this is one of the first single molecule measurement of within an image, which has evolved over multiple different atlases and measurements across, like, hey, now we're able to do it this species, we're not able to do it this specific genes, how many genes and different types of RNAs, which had all the way become like Allen Brain Atlas, one of the very first atlas efforts, where they were trying to do, hey, we're doing one RNA at a time or one-ish at a time. We're going to document everything into an image and start creating a database. And then moving forward, people now started developing different technologies. Sequencing has come on, and also different microscope technologies has come on. So the very first thing was Baker's dissection methods, where you can have specific regions and you tag these specific regions of interest, and using the laser or any kind of like UV cleavage methods, cut out these specific regions and take it as more of a regions of interest. And this is a very first form of spatial transcriptomics, not necessarily a single cell or single molecule measurements, but now we're able to specify the regions that we want to understand and only select that tumor, only select that specific crypts, and collect them for sequencing. This was a very first form, which has also evolved into a fish side as well, where you try to now measure, not just doing sequencing, but also on the imaging side, trying to do multiple measurements at the same time. So this was the very first form, where they're trying to visualize multiple probes with multiple fluorescent tags and try to do a cyclic imaging, so that we can do multiple, more than 5 to 6 genes at a time, but as many genes as possible as we go on to the higher plexing. And 2014, 2015 is the one we first really talked about imaging-based spatial transcriptomics, when the seqfish and merfish came along, which we'll kind of discuss this in more in depth as we discuss the imaging-based transcriptomics. And 2016 was the first year when they're first starting to talk about, okay, we're defining the term spatial transcriptomics, uh, which went on. And fast forward 2021 and 2024, as I mentioned in the first class, they end up becoming a method of the year. People were very interested. There's a lot of publications around it. And 4 or 5 years or less than a decade down the line, we are now seeing a plethora of different companies or different platforms or different approaches to meet the needs for your research studies, really understanding how high we can go in terms of multiplexing capacity, but also how high of a resolution can we go for, or how granular we can go with subcellular resolution imaging technologies and sequencing as well. And I like to always think about technologies are cool and we can do so many different things, but also why we're doing it. And the very fundamental reason is that every organ has a spatial organization. So you're seeing liver as an example where we can see the zonation patterns. You can see that they're going from like central vein to portal vein. There are specific cells organized in a like hexagonal way that you define as a module. And if you just look at H&E, it's not as evident as it should be. When you start to stain them and color them in different colors, you're now starting to look at these tissues and understand it a different way, like where are the portal veins located at? What are the cell types? What are the microenvironment that they are part of? Like, are they exactly the same if you're looking at two different portal veins? So these kind of like spatial organizations is what really makes it cool to have these high resolution imaging or multiplex FISH or sequencing into play, because not just the spatial organization is important, every biological processes undergo these spatially localized changes. You're seeing liver fibrosis development that starts from vasculatures and going all the way down to all the way to epithelial layers. Also the wound healing and scar formation processes, where you can see during the wound healing processes, there are different gene expression programs that are getting turned on and off on a very specific regions from the surface to the bottom, that we oftentimes cannot capture if we were to just do bulk RNA sequencing and hoping we're seeing upregulation of specific genes. And with that said, this is also very relevant clinically as we are going through, everyone calls precision medicine or digital pathology, where it started all the way down from histopathology, where we try to look at the structure of the image of the tumor sample. If the patient comes in, you take the H&E, you take the biopsy, you take the H&E, you maybe do immunohistochemistry on like a couple of markers to see, hey, is bracket like HER2 positive, HER2 negative, and or whether this tumor is stage 2 cancer or stage 3 cancer. And then we develop as we develop different technologies like next generation sequencing, PCR, or molecular FISH, to do the companion analysis. We're now more than able to subtype the patients, not just to their like staging of how many tumor cells they have that are evident in their own structure, but also what kind of molecular subtypes that they belong to, what kind of treatments these patients will respond better over the other. But now as we're merging these two together, we're not doing one sample, one biopsy into H&E and one biopsy into next generation sequencing. With the spatial multiomics, we're now able to do on the same sample H&E and sequencing on the exact same block or exact same site that we have, which allows us to integrate everything that we're seeing and locate that this specific cell is the one that is resistant to treatments, or truly personalizing all of these different informations or different omics layers that are curated to that patients. And we're currently undergoing and understanding a lot what what we're seeing right now, honestly, or what we can do with this depth of information that we can get out of a single patient, and going towards like a true digital pathology. And this is, I always like to mention it because this is exactly why technology is necessary, not because it's cool and because we have, of course, that computational biologists or data scientists would love for any more data, but this is why it creates a clinical impact, not just a coolness technology, but the information that we're getting out of it. With that, even if we just say spatial transcriptomics or spatial proteomics, there are actually, as I mentioned, a lot of different technologies that are out there. And when it first came out, like when it first started taking a lot of publication or attention around 2020, 2021, we tried to put together a review paper, which got published in 2022, really understanding what kind of technologies are out there, what kind of considerations and what kind of analysis tools are out there, and put together a comprehensive review on spatial omics technology in general, which I think more than half of it is outdated at this point, but there's still a really good insight if you want to take a look at the paper. But the main points that I'm putting one of the figure over here is we can broadly divide spatial transcriptomics into two different categories. One into more of an imaging base, which is oftentimes more targeted, like where we're using antibodies or probe-based panels and libraries to hybridize onto the tissue and do the readouts. And the other option is to do transcriptome-wide using more of a sequencing-based methods where we can do in situ capture of all of the RNAs or all of the mRNAs and do the sequencing. But we're doing more of an untargeted analysis where, although we might be compromising a little bit on the capture efficiency and rates. So the way I structured the next hour or so is to really go through these contents and different technologies to consider if you really want to run spatial omics, like what kind of questions you should be asking yourself. And of course, the very first question, which I want to ask you guys, is why do you need spatial omics for your project? Because a lot of the biological questions can be done in multiple layers, right? It can be histological, or you can take a lot of the hepatoxylin and iodine staining and look at where's the tumor, where's the invasion, where's the immune infiltration happening. You can go all the way down to cellular, you can run single cell or single cell on what are the cell types that you're interested in. Are you interested in immune cells? Are you interested in fibroblasts? Are you interested in like different tumor microenvironments, different cell combinations or subtypes at the same time? Or is it more of a molecular of like, it can be viral infection of like specific gene expressions that are going in and out, or specific mutations, point mutations that are happening in specific regions and specific cells that you need to really pinpoint the specific molecules playing around or spreading around within the tissue or within the models. So I want to pause here and ask, like, how many of you guys have run or are considering running spatial? 1, 2, 3. And for you guys, like, why do you guys need, or why did you guys get interested or need spatial omics for your own projects?
For myself, those are kind of two different things. Like when I was first exposed to it and how I'm thinking about it might be utilized, like in search now. But some of the questions that my own research would depend upon is markers of neuronal populations and the differences between them, so that I'm able to use like a strategy that's able to differentiate like one population versus another, which is difficult to bring because there's so much overlap. And the brain region I'm interested in animal model that I'm interested in, it's just not well defined, and that has been very well characterized. So I think it would give like a really big picture of like these different gradients and like proliferating like progenitor zone to like one more mature population, what that actually looks like. So age is kind of how that would change from exiting the progenitor cycle and longer proliferating to actually reach maturity. So seeing spatially that's maintained. So I think
Okay.
Yeah.
So it's more than a simple RNA trajectory, but also spatial trajectory.
Yeah.
Any other?
The part that I'm working on the it's on premalignant cancer patients. So the reads are very, very small, so it reaches to like a single cell spatial, and like such a small number of cells.
Yeah, I think structure might have to be defined a little further, but that's a generally good big project. And lastly, last but not least, what was the ovary?
Yeah,
Yeah.
so there's like follicles, like a bunch of different follicles in the ovary, and they all have like an oocyte. And so if you want to look at the transcriptome of each cell, like if you do single cell, you don't know which follicle they belong to. So if you want to see like which follicle is going to be the one that releases the oocyte, you need spatial to like segment everything and then compare.
Interesting. Yeah, so a lot of the questions that you guys had was around cellular, it seems like. And there is a lot of consideration, especially because how I personally also got started spatial was to identify the cell types that cannot be identified in single cell, and to locate them to define where they are and find what they really are, which had its own challenges in terms of like identifying the right panels, but also if you can segment them properly and understand, find these cells in the first place in your data sets. So hopefully by the end of this lecture, you will be able to get some of the answers, some of the solutions that you might, all the problems that you might have, but feel free to ask questions if there's anything that could help your own projects. And this is where you're starting, right? I already asked questions, can I use spatial transcriptomics or is spatial be any useful to you? And then you now questioned around like, what's my experiment like why do you need these kind of information? And a lot of the cases, it's either hypothesis generating or hypothesis testing, where we oftentimes ask, it's now less about whether you want to generate or test the hypothesis, more of like, is the cell types or questions that you have being like can be defined within the markers or within the set biomarkers that is already in the panel, or that can you optimize these antibodies or probes for your cell types or for your question or pathways of your interest. So if you already have a well-defined panels or a list of targets that you're interested in that overlaps, you often go with imaging-based methods where we can do the imaging, the small molecule FISH or multiplex IF to specifically identify these markers plus other cell type specific markers. That way there is a little less background or a higher sensitivity of the markers that we're interested in than sequencing, especially if you're interested in rare cell types. Sometimes they express very low levels of RNA and have biases when you do the sequencing. On the other hand, if you're generally looking for, hey, I want to define microenvironment of this specific tumor or this specific diseases, or if you're still looking for markers or clusters that you want to specify, you often tend to go with sequencing-based methods. The benefit of that is untargeted, you can go for whole transcriptome, but you may be compromising either resolution or capture rate a little bit when we actually compare to the imaging-based analysis. And then there's now a series of questions of like how we can gather images, how we can segment them, like how easy it is to preserve the samples, and these kind of questions is more an experimental side. So this is a spatial omics workflow in a very simplified sense. You prepare a tissue sample, you trap them, image them to see which regions, if you're covering the regions that you're interested in, do the library construction, either for sequencing library, or you can hybridize the panels or stain the panels for your spatial transcriptomics, sequencing or imaging acquisition, and then data processing and visualization. And these are like a million questions that you can still ask every single step, which hopefully you can have a little bit of answer today, or at least how the field is figuring out right now. So starting with the sample prep itself, like you already have specific samples that you're interested in, but there's still a lot of questions of like, hey, are you getting the samples from biobank? If so, or from your own experiments? If so, what kind of preservation method you have, how big they are, how big you should go for any imaging, what are the tissue types, and also, more importantly, is the structure more important or RNA quality more important to you? So it's first step is to save your samples, like fixation. And a lot of you are familiar, if you guys are doing lab biology, there are two different ways, right? You can do physical preservation by freezing them, or even there are a few different ways you can collect the information from these samples, but freezing is a typical way for any kind of like tissue samples from biopsy surgeries or experiments. Chemical methods, there are a couple of different ways. When it comes to the frozen samples, we tend to use methanol fixation or alcohol to dehydrate and fix the tissue structures. More commonly, we use aldehydes, like 4% paraformaldehyde to cross-link everything on the tissue to keep the structures, but also keep the RNA and protein information in the tissue. And there are tons of different ways. There are also reversible cross-linking. There are different chemicals you can use. There are different fixation protocols that we can use. But I think a bigger question that you always have to ask when you do spatial is when you fix the samples and when you keep the samples, how uniform and homogeneous will they be? When it comes to, for example, when you have a biopsy or surgical resections from the clinics and you want to do a clinical study of like colorectal cancer patients, we don't oftentimes have a control over how long they're sitting on pathology labs or how long they're fixed for. And when you run spatial on these guys, you can see the overfixations on the surfaces of this tissue and underfixation in the middle of the tissue. I think that very similar things can happen because some regions are very dense and high in fibroblasts that has a lower division rates. And some regions are just well penetrated and can be easily fixed. And although there is a lot of fixation protocols out there, and there's like, if you go to like pathology labs or if you go to different labs, you can just say, yeah, for any RNA fish, you can do like 20 minutes fixation or whatever, however big or small your cells or tissues are. You have a set protocol, but always make sure you are not overfixing or underfixing the samples, or else it's going to be the errors that you're getting is far bigger and higher resolution technology because it propagates over time. And also the temperature as well. Sometimes when you go into surgery, they tend to put the tissues onto the ice or room temperature. And what we learned over time was it actually impacted the RNA quality that we had on a spatial transcriptomics, which we didn't know from any of the immunohistochemistry or immunofluorescence stainings or even the RNA scope runs that we had. And of course, the concentration and protocol and the pH matters, especially when it comes to the sensitive tissues like digestive tissues that has a high enzymatic activities tend to degrade faster than the other organs. Brain has a lot of RNA, whereas like there are organs that like pancreas that will lose RNA over time very quickly. So these are some of the considerations and research that you have to do even if you have really full samples or cohorts available. And similarly with preservation as well, like sometimes you don't have a choice over this, but oftentimes if you're running the experiment or if you have it, if you're collecting the samples yourself, you can either choose to do FFPE or flash frozen tissues. The verdict is if you want the RNA quality to be pristine, flash frozen tends to give you a better quality. Just because you're not fixing and masking some of the regions of the tissue. But you will have, because you're sectioning at less than 10 micron every time you're doing spatial transcriptomics, it's inevitable for you to have a little bit of freeze-thaw cycles. It's really easy to tear and lose the structures, especially if you're interested in how structurally the tissue is changing. But on the other hand, FFP tends to have a really good structure. You can always go back to 3 to 4 year old or even further. There was a very interesting paper in Spatial that they went back to like 100 years before like Spanish flu tissues that were in formalin and run spatial transcriptomics. So FFP is a really easy way to store and go for chival tissues and run spatial transcriptomics and have a really good structures. Although it's not going to be as good your RNA quality. Or if you're very interested in like cells that are very sensitive to this type of a degradation, FFP not be the best preservation method that you want to have. So that there is a bunch pros and cons, really depending on your biological questions. And even if you fix them in FFP for formalin for frozen like methanol fixation, you also have a second step of like how you permeabilize and retrieve the antigens for like antibody-based methods. And different, like when it comes to bone, you need to consider about decalcifications. If it's brain, sometimes people do tissue clearing. So there are different considerations that does inhibit the detection of the RNA, which is also one of the considerations that you want to make before you choose the platform. That even if it gives you a highest amount of like, very first mistake I made in spatial transcriptomics, I went for a whole transcriptome method at highest resolution possible. What ended up happening was it was only available for flash frozen and the structure was completely messed up, but also the capture rate wasn't high as I wanted to be and might have been better just going a little lower on the plex or a number of panels, a number of genes that I can detect at a time, but rather make sure I have all the structures and cell types annotated properly. So yeah, these are some of the very first questions you might ask when it comes to your samples itself. And these are very common questions when we collaborate with people, or if anyone wants to go into spatial and have like, hey, we have this really cool samples or cool projects that we want to run spatial transcriptomics or proteomics on. Is there any questions or anything? if not, now we can consider, now that we have a properly stored sample, we can now consider, okay, what kind of technologies do we want to use? What are, yes.
Actually, sorry, I do have a quick question.
Mhm.
I was wondering if in your experience, what tissues tend to work better or worse with spatial transcriptomics platforms? Like, I know that people keep trying to do bone marrow spatial transcriptomics, but it doesn't always work out. I'm just curious.
Yes, bone marrow was a huge challenge. There, I think the typical tissues that we have a lot of data on for spatial transcriptomics, like pollen, for example, has been not only it's really easy to get good quality samples. It's one reason why we're getting really good data, but it's also very structurally defined. And once it becomes FOP, it's really well preserved, and that's one of the most successful data. People have done a lot of lumps too. The downside of that is you actually waste a lot of space because half of them are just air and empty space with a lot of airway. But the general, like, I think COVID actually helped optimizing a lot of protocols. We've also seen a lot of tonsil data where there's a lot of immune cells and a lot of activities. So I think these are some of the tissues that worked really well. When it comes, tumor samples tend to work really well because they have a lot of RNAs and they have great, like, they're proliferating, they're like active with like all the immune cells, so they're lighting up perfect. Whereas this was really interesting because pancreatic cancer had the exact same issue. Like, we had a beautiful pancreatic cancer data sets, but our control tend to have a lot of RNA degradation just because they were more metabolically active at the time of collection. It also depends on what you're, like, how you're collecting them. Surgical resections tend to be better unless they're not been sitting on surgical surgery room for like overnight. A lot of surgical resections were giving us beautiful data sets, whereas biopsies tend to have a little bit of variations in quality. Bone marrow has been a very interesting, there has been a lot of people who wanted to get the bone marrow done. I think the decalcification protocol has now been figured out to a certain point, but you do sacrifice quite a bit of an RNA quality. So we tried like aged bone marrows and like people with high ages or people with like inflammation. They tend to fail badly, and we're now currently trying like different methods like pseudoblacks to reduce the background or different ways to kind of get around that. But like bone marrow is one of the interesting samples. The normal samples tend to do better than the diseased. But I think it really depends on. I think there is an organ that tends to do better. Brain has been one of the best child of the spatial proteomics, spatial transcriptomics, just because the amount of RNA is tremendous. Segmentation was hard, but it was one of the first spatial omics that's out there. But when it comes to these kind of like more tricky organs, I think the protocols being trying to get, but then there is also a mixture of like protocol, but the sample quality itself. I think that's what made it really hard for us to define what's easy and hard in the first place. Because there are tissues that are really good in quality but has high in variation that makes us hard to analyze in the first place. COVID gave a little bit of an overview of like how everything.
Yeah, that makes a lot of sense. Thank you.
Yeah, and going on to different platforms, I think you've seen the slides before, where these are available platforms to consider if you were to do spatial omics in Cornell. We might actually have more, but these are one of the major ones that we have. Through Cornell, if you were to run it, there are a few machines all around the Cornell MSK Rockefeller campus that you can consider, which are all like part of different types, like Cosmics is one of the Cosmics and Xenium is more of like fish-based methods, whereas Cellscape and IMCs are more antibody-based, whereas Cellscape is imaging and IMC is more of a mass spec. But there are like, and Visium and StereoSeq are more of a sequencing-based methods, and Pixelpin is so it really depends on the questions, and hopefully this gives a little bit of an answer throughout as we discuss one platform by another. So going on to the first part where we talked about imaging-based spatial transcriptomics, where I first talked about the very first like single molecule imaging of ribosomal RNA and messenger RNA. So that's a basic technology that they're relying on. There are two different types where people call in situ sequencing, where they're hybridizing. So the concept is the same. You have a specific probes designed to target specific regions in your cell or within the tissue, and you bind these specific mRNA probes onto the tissue surface. And the difference is the detection, right? In situ sequencing involves more of amplifying the signal through rolling cycle amplification, which basically is amplifying the signal after you bind this mRNA probes. Or in situ hybridization is rather than amplifying the signal, you add additional probes or fluorescent tags or any kind of detection probes to amplify the signal and detect them or understanding their spatial localization. And there are, so that kind of divides into like more of a targeted, untargeted, and fluorescent. So the first two are more of a in situ sequencing where you have an amplification, and FISH is more of like how you play around with different barcoding methods to amplify the signal of a detection. So starting like the very first step is, as I mentioned, you binding the mRNA probes onto the specific tissues or specific regions within the cell. And the concept itself is very simple, right? You have a cell that are expressing specific gene, and you want to target that regions with the right complement sequence that has like fluorescent tag on it. The challenge is to get to the spatial transcriptomics or like omics level analysis is like how will you multiplex these 10,000 genes onto a same within the same cells or same species? And two, if you start multiplexing, yes, even if you can do like 1,000 genes at the same time or develop 1,000 probes and bind it all at the same time, are you gonna physically fit that onto the tissue surface because you're adding the probes and adding it on top of the physically restricted space? And one big way in terms of like FISH-based methods is to use the barcoding strategy. And it's been evolved slightly differently as they are coming up with different ways to barcode. Like SeqFISH was the very first FISH or spatial transcriptomics where they are trying to have multiple fluorescent probes and almost forming a barcodes by linear probes. It was good, but the signal was still very weak because it was every place it binds was a single fluorescent tag. And in order to amplify the signal, they developed more of a sim HDR of like changing the probe shape of the probes to add more fluorescent tags. And the downside of these were more of like any off-target effects and non-specific bindings as they increase the flex of these technologies. And that's when the MERFISH came out and discussed, hey, we want to define each of the probes as a form of a barcode, essentially. So in a probe, they will put multiple fluorescent tags that are representing on and off, which is like 1 and 0, so that each of the genes will almost become like a barcode of like 11001. For each of the genes, you will have those individual barcodes and detect them. And by having a barcode and they will add empty barcodes for error correcting methods so that you can have better signal, but also avoid to a certain degree like false positive and false negatives as much as possible. So MERFISH was the very first technology after all became commercially available, which now is a form of a Marsco. And this is one of the kind of a groundbreaking technology that a lot of people talk about these days in terms of and how they came up with the idea of not just the probes that are targeting specific regions, but making those probes as barcodes so that we can capture them and quantify them without a lot of errors, as if we were doing a regular FISH in a high multiplexity way. MERFISH was the first technology that went all the way up to 10,000 probes in spatial transcriptomics, and now people are going through like now it's on surface and you're able to capture whole transcriptome with a fish-based methods. And Seqfish 2, like Plus, is more of a similar to Merfish approach, but in a very in a Seqfish backbone. So they're basically trying to have specific probes and similar barcoding scheme, but more targeted to a gene. So this way they can do higher plex because they're using bigger probe sequences similar to SIMH small molecule ICR, but still limited in terms of a capture efficiency compared to other antibodies or sequencing-based methods. And that's when people came up with in situ sequencing, because even if you take these different barcoding methods to detect and amplify the fluorescence signal, there is a limitations to which you can really get those amplified. So how it differed is you're using in situ sequencing. You're basically taking the same approach of binding the probes, but you're getting additional padlock probes so that it increases specificity of you can double bind to make sure it's bound to the right target, but also use this as a template to amplify and have this kind of a ball or multiple circular structures where you can where we call it as a rolling circle amplification to maximize the signal and visualize in an imaging in a more abundant way, essentially. So this is one of the very backward technology of xenium technology, essentially. But there's also a lot of different technologies that came out that are using a similar concept, but using a slightly different way. I don't have it here, but and also not just the way you're designing the probes, there are slight different improvements in the workflow that came out and different variation. Expansion sequencing was to overcome the crowding issues that all of the technologies had, where you're cross-linking everything, embedding in a hydrogel, expand it so that you can have more probes bound to within the given area. There are a few different ways people develop like snail probes so that they can use like enzymatic reactions to amplify the signals, or fish-seq where you're using more of a different probe, like fluorescent probe designs to go for like higher multiplexing capacities. Although there's, but after all of these, I think a lot of the verdicts that we had, which we're still suffering right now, is as you go higher multiplex platforms, you tend to go lower efficiency, or you tend to like have a higher false negative and false positive probe binding events, which is still quite a bit of an issue in the imaging-based spatial transcriptomics side. And I think there are I think ISS is more of a backbone for xenium. The small molecule fish is a backbone for cosmics, and we have multiple, and Marfish as well. We have multiple technologies that are commercially available that are working really well, asking the good biological questions. And this is the most recent advancements that's going, is more like, hey, can we use this same methods and go all the way up to 3D? I think Cosmics went all the way up to whole transcriptome, around 19,000 probes at the same time within the same sample with the probe-based technologies. And now people are looking into not just the whole transcriptome and increasing plex, but also going for 3D where you can clear the tissue structures and try to do embed in a hydrogel and doing the same probe technologies to do the 3D imaging. And this is more of a, one of the examples that we saw recently at HMP conference, which is coming in a couple of years in commercial regions. And yeah, one of the exciting advancements, we're not just looking into a 2D base, but starting to look into 3D structures of these spatial transcriptomics data sets.
Can I ask about the 3D?
Yes.
Is it, sorry if I missed it, is it that they're maintaining the three-dimensional structure, or there's like if you're able to give the 3D tissue and then it will slice and segment?
So for this specific, what people are doing currently right now is to have consecutive sections and reconstructing 3D structures. For this one, they're taking a thicker section, I think up to 550 micrometers, or I think even more. and embed them in a hydrogel. It's almost like a lump of the tissue, and of course, you have to optically clear them. But then once you clear them, and do the having enough like calculations on the penetration of the probes, you're basically applying the same concept. You're doing light sheet microscopy on these thick tissues after attaching these spatial if hundreds or maybe like thousands gene-plex spatial omics library. And the next part is also imaging based, but more on spatial proteomics side, where sometimes the RNA is not of your questions, of you're more interested in how it's getting translated and phenotyped in protein layers, but also there's a lot of applications in pharma side where they're interested in biomarkers and already have an optimized antibodies that they want to really define antibodies for specific cell types or subtypes that you're interested in delving into. And that's when they use a lot of these imaging-based proteomics methods. The very backbone of the technology is also very common molecular biology technology that we have in our lab, immunostaining, immunofluorescence. I'm sure a lot of you already know how this works. You basically have tissues and cells, and each cell has its own antigen, and you're basically using antibodies to detect these specific antigens to detect them. And you're using secondary, or sometimes we use a conjugate that already has a detection antibody where you have enzymes. You can digest the substrate and visualize it, but you can also add a fluorescent tag. And once you visualize them, you have this image of each where the cell is, but also where these proteins or where these antigens are presented at. And it's a really simple, really common technologies, although it's hard to master it. It's really funny when you go to pathologists who basically some of the staining results, depending on what you get, the subtype of a tumor or subtype of disease can change for him. So sometimes there is a difference in level of perfection you can have. One complications when you always deal with the spatial proteomics, but also every antibody takes a lot of optimization, and you have to consider the spectral overlap of different fluorophores you're taking. So that's been one of the big challenges from just having a 5-marker staining in a paper versus going all the way down to multiple hundreds and thousands of markers. Is one spectral overlap that they have, but also the antibodies tend to cross-react and have a non-specific staining. And these are some of the challenges that they had to really bring down to having a 1 or 2 marker staining to multiple markers and really call it spatial proteomics. And of course, because a lot of these are fluorescence, once it becomes liver or even brain, where tissues tend to autofluorescence a little bit, tend to be a little bit of an issue, especially when it comes to GFP or specific fluorophore channels, which is now can be overcome with chemical and different photobleaching methods, and also there are a few mass spec based methods that I'll introduce later in this part. So I think to overcome and go all the way to multiplex methods, the immediate solution was remove the tags or fluorescence tags after use. So the basic concept is very similar. If you can image 4 to 5 markers at a time, we're going to do it cyclically so that we can have a stacked image and call it spatial proteomics. That was the very first cyclic IF, where you can do fluorescent label imaging, and then you remove the antibodies by chemically stripping it, and then went all the way over. The benefit of that is you can get 40, 50 markers easily. When you look at the tissues, every cycle you can see the tissues are getting brutally torn, essentially, because you're stripping them over and over again. So if you're dealing with very sensitive antigens or very transient phenotype, you might want to put it in the very beginning of the cycle, or you might not be able to detect it at all. And with this, I think that's why people started to question, what's the best way, or what's the most gentle way to remove the signal without too much of a spillover or contamination across the cycles. So the very first few methods that people used were fluorophore inactivation. by using like hydrogen peroxide or other reagents. You can use alkaline oxidation to remove some of the detection probes. And chemical bleaching was one of the common methods in a lot of these spatial proteomics methods, that you can just um, strip the antibodies and run an extra cycle. The benefit of that is you will not have a lot of backgrounds, but then you will see, as I mentioned, every cycle will be will see the degradation of the tissue. A lot of technologies use photo bleaching, by taking doing photo bleaching the fluorophore essentially. So you're keeping all of the antibodies and detection probes, but you're basically killing the fluorescent probes and adding another ones or sets of probes in another cycle. And there's also now developed antibody elution buffers, which is very similar to chemical bleaching, but a little gentler to elute the antibodies and doing the stainings again. So these are more of like different strategies. People started like removing the antibodies, but now, or photo bleaching it, but now started to like, maybe we can add just remove just the fluorophore. We can start to do more of like different components to see how much we can gently remove these probes or detection sets, but without compromising too much on the sample quality. And as it kind of developed, people started to add more than fluorescent tags because of the backgrounds, but also because it was very limited to what you can do to remove the signal. So they started adding DNA reporter tags on top of the detection antibodies or antibodies to detect, instead of having a detection antibodies or with the fluorophore, you're going to have a DNA reporter tag that is already pre-attached to the antibodies that becomes very similar to the imaging-based spatial transcriptomics that we just discussed, but still you're detecting the probes with the antibodies. So, and then once it becomes, you can strip these specific regions of the DNA reporter tags with photo cleavage, or most of the time photo cleavage and with the UV, and going over the cycles to develop. And this is the backbone technology of Codex now, Cycler. The benefit of that is you can develop, you can have higher multiplexity without a lot of sacrifice or a lot of consideration. The downside is you do have to generate your own DNA barcoded antibody to use pre-optimized sets and have a less amount of customization. And that's when the more of a technologies like IMCs or MIDI, if you've heard of it, came out. Is rather than using DNA reporters, we're going to take mass spec or imaging them in the first place to deal with the autofluorescence or background issue. We're going to use the mass spectrometry to read what's in there, essentially. So the backbone is the same. You're still using antibodies, but now you're either metal tagged or you can have different tagging methods and ablate every micron of this tissues and run it through mass spec. And the result is every pixel you have will have its own mass spec results, and you can reconstruct it back to more of an imaging form depending with that metal tag mass that was detected in that specific pixel. So the benefit is you can multiplex them and you can easily make the metal tag antibodies. And with doing this with still doing the spatial proteomics without these downside of the fluorescent based methods. But then the downside of that is the optimizing the metals or understanding how metals behave has been its own expertise required field, and also understanding the mass spec as well. But the resolution cannot go lower than 1 micron since we're using the laser to go every single pixels. And also lastly, the cost of the metal, some of the inert gases or metal type antibodies can be expensive or very harmful to the environment because we're using them and essentially throwing them out after measuring them. There are also like methods like MIDI, which is very similar to this concept. That's why I didn't put another slide, but using the ion beam cryptography. But this is exactly the same concept that was applied in metabolomics as well. If you're interested in spatial metabolomics, it's a very similar idea of like ion beam or mass spec. You can use these specific regions to not measuring the metal tags, but measuring the metabolites after you treat them. So there are tools like platforms like MALDI, DASI, SIMS that sometimes people do spatial metabolomics or doing multi-omics with the spatial proteomics, but the resolution generally is lower, but you can multiplex them or you can run consecutive slides on top of each other to do multi-omics on proteome metabolome, which has been quite popular in digestive diseases and GI research. So these are more of a summary of like, hey, we have antibodies, we're still using antibodies, but we're using different ways to strip the detection probes or develop different ways to read them or detect them. And I think there are small different technologies here and there that are developed, like Immunosaver, for example, has different ways to develop a probe. So there are, but then the concept itself is the same. You're still dealing with specific antibodies or specific clones of the antibodies that you need to choose and curate for your samples, which can be really good if you already have optimized, which can be a little challenging if you don't have, if you're working with a subtypes of the cell that is not well defined in the field. So again, with the multi-omics now, we're not just proteome metabolomics, as I mentioned, we're also going towards not just stuff from proteomics because of that exact challenges but then to add additional RNA in a form of like RNA-scope, RNA probes, or even for like sequencing, that you can do few stains of a proteomics using similar these platforms that I mentioned and run RNA consecutively because you can strip every antibodies off with this technology. So even if you have a little bit of background, you can still have an RNA layer and try to integrate them, which I'm going to show like towards the end of the more of an analysis part of this lecture. So yeah, this is an example of like spatial metabolomics and proteomics that I mentioned with using SIMS and proteome. I think it was SciMC, but imaging-based method, like spatial proteomics methods, and you can almost have different layers of like metabolites, nuclei imagings, and proteomics, and do multi-omics studies altogether. So this has been one of like an outlook of how we can multiplex or the modalities of different spatial omics layers within the imaging space. That's been one of the big advances that we've been seeing for the past couple of years. And lastly, on proteomics side, this is very interesting technology that is not really a traditional proteomics measurement that I mentioned, but we're now going all the way down to not just where the antigen exists in the cell, but they're also going all the way down to subcellular, where you're trying to now ask about how they're pixelated, which they call as molecular pixelation, but show how the proteins are colocalized in space. So basically they do these antibody binding and then fix everything and do the detect these specific antibodies and do the imaging and do the sequencing consecutively after the library prep, which really allows them to understand not just how much proteins on the cell, but also what are the colocalization of, like, what are the probabilities of these two antibodies that are bound together in a very similar space within the cell. So you're getting all the way down to subcellular level and seeing which markers are colocalized together on the cell, which I'll show the real world data towards the end as well. And the last part of the platforms, after talking about like at least 50 different methods, is also sequencing-based spatial transcriptomics, where the differentiation factor is we're not necessarily using imaging, we're using ultimately sequencing, which involves library prep and sequencing. So if you're having any targets or any questions that you want to avoid the sequencing bias, this might not be the best platform. But the benefit is you're going for unbiased. You can do any sequencing level analysis of like nucleotide level analysis on these data sets. The way it's achieved is you're embedding all the spatial information onto barcodes. So it's find without any imaging, although now people tend to do at least nuclei imaging to map it back into the, or segment the tissues. Whereas like molecular information is all stored in the sequencing library. There are a few different ways. A lot of the cases it also varies by how it's captured. So barcoding, like where the barcoding happens or how the barcodes get retrieved is what makes these different technologies different from each other. So there are three different ways, right? The first one, which I'm probably gonna really talk about it briefly, is LCM based. You can, as I mentioned towards the beginning, it's a classical technology where you can use a laser to capture specific regions that you want to have ablated and collect them for sequencing. But what's more common now is more of an mRNA capture onto the slide. So you can have the pre-recorded, can be false beads or barcoded space, or probes that are organized in like circles or squares, and you can permeabilize tissue on top of these barcoded surface and collect these and run the reverse transcriptase and run the cDNA synthesis, and you sequence the cDNA to get the spatial information or spatial transcriptomics onto from these tissues. Last one is more the microfluidic space, where you're using you're first fixing the tissues and permeabilizing and barcoding by attaching a microfluidics device on top of this tissue, which is slightly different from what most common way to do it. So the very first version of spatial sequencing, so to speak, was from 2016, where they came up with a way. Let's say we have different like 100 millimeter tiny balls of regions where you attach different probes on top of probes on these barcoded space, and each of the spots will become barcodes. You can have the different IDs on top of them and put the tissues. You can permeabilize it so that these regions that were sitting on top of this specific barcodes become more of a spatial barcode that you can map it back later. And this was the very first concept where it was very clearly you were barely getting like valid nature sequencing sequence, which was around very similar to single cell back at some time. And you're getting like 100 and 200 micron resolution across very small amount of surface. And you basically have each of the barcodes had a cleavage site where you can retrieve the libraries or reverse transcribed and cDNA synthesized libraries, but also the amplification sequencing handle barcodes, and the rest of it becomes really similar to any single cell methods. And which basically became a Visium standard definition or regular Visium after commercialization. So there has been quite a bit of optimization that they ended up from original paper, increased the capture rate and resolution, but it was still 55 micron diameter technologies where you're seeing a grid level or gradient of expression patterns. But one of the breakthrough for Visium technology is cytosis, where you can have the sample prep, like you can prep the tissue, then you don't have to, you can buy the slides and do it more of a conservative workflow for these spatial technologies. And that's probably why, even if it's a really low resolution, and I was at some point joking around, it was really spatial because you can't really you're getting like 5 to 10 cells within this 55 micron diameters with the gap especially. But still, people have generated so much data sets on Visium because the workflow was easy enough to implement. And at that similar time, there was SlideSeq, which later got commercialized with Curio, but it was a similar concept. Instead of spatially barcoded regions, they call it index beads. So you're basically putting beads on top of the slides, and you're retrieving the beads without really making a cleavage site on the probes. So you have a beads onto the slide already. You have the same process as a Visium where you put the tissue on top and permeabilize it, get all the information, and do the library prep and get the sequencing out. And you can, by each of the beads will become one barcode, and you can map that to spatial locations. this allowed us to get the beat, because of the beat sizes, you can get up to 10 micron beats, which was the commercialized version for Curio Seeker, and which later became Curio Tracker, which they came up with more of a slightly smaller beats, but also they allowed the beats to detect specific regions in the nuclei, so that it more became a single nuclei sequencing with the beats. So every regions that the tissue's nuclei was sitting on top of the beats were collected and cleaved, and after library preps, you can almost have a single nuclear RNA sequencing, but with the spatial barcode information with it. The benefit of that is you don't need a full equipment like Visium, Xenium, or other spatial transcriptomics and proteomics methods, and it's relatively simple because you literally have beats and getting prepped, and the rest of it becomes really similar to your single cell workflow. But when I first saw this tracker technologies, immediate question that I was is like, how much do we have to sequence? Typical single nuclear RNA sequencing takes 10,000 cells at the maximum, and if you were to do this, you will take multiple libraries to cover all of the regions that you want to cover. And currently, it's only available in flash frozen tissues. But if the sequencing cost is not an issue, I think this is a really strong technology, as you can go all the way down to a single cell level, but on the whole transcriptome size. There has been a lot of push in terms of like higher resolution, especially after I talked about like 55 micron and 10 micron is not enough for a true single cell. People I think there were actually original developers of Visium use the split pool chemistry where you're basically barcoding, and every round you're adding information so that you can have multiple combinations without having all of these barcodes like pre-allocated, and which has become a backbone for Visium HD. So even if there are called Visium and Visium HD, they're almost completely different technologies because the way they're capturing is slightly different, or the way barcodes are generated is completely different. And it almost allowed us to go all the way down to 2 micron resolution. So this is how the Visium regular or standard definition would look like at 55 micron. And now with Visium HD, you're going all the way down to 2 micron, where you can, it's a level of subcellular resolution, although most of the time you bin them at 8 micron resolution bins to do the analysis. It's still allowed to do somewhat of a subcellular analysis, unlike other technologies on transcriptomics or sequencing-based technologies. And now that people are pushing for higher resolution because they can do whole transcriptome already, there are now more methods that are sub-micron spatial transcriptomics. One example is StereoSeq. It's the same concept. They're basically using flow cell and make the DNA nanoballs, which allows them to have smaller, which becomes like 220 nanometer resolution, and do the spatial sequencing. And the resolution is highest in the market. The capture rate was slightly lower, and you ended up binning them anyways to get the right coverage. But still in one of the very unique products in the market that is, depending on your question, might be worthwhile to try. Illumina has recently, I think it was in February, they announced that Illumina Spatial, which is based off from a SeqScope technology that was already published, also using their own flow cells and using their own probes to put the tissue on top of these flow cells and doing the library prep, and using their sequencing platform to get the spatial. And you're talking about similar 2 micron resolution on these datasets as well. The specific details on how the science works behind it is still pre-commercial and not been fully uncovered. But this is one of the example ways that everyone has Illumina, or a lot of the institutions have Illumina sequencers, and you can pretty much use their flow cells to start running spatial. Was the groundbreaking part of this announcement. And speaking of the sequencers now, like there are a few different additional players in the market that are using sequencing-based technologies, but more like on targeted sequencing, not so much on untargeted whole transcriptome. One example is an element of BD24, which are allowing you to do more of a multi-omics on the sequencer. You're essentially using the flow cell, you attach the cells, or you prep the cells and you attach the flow cell components, and you can do the imaging of a proteomics or antibody staining before you do the sequencing, and then you can do targeted sequencing after that. Really allowing you to do multi-omics at the same time. If you want to do like CRISPR screens, you can now do it with this. And singular genomics G4X as well. They're also using their own flow cells. They're using slight changes in how you're embedding tissues, but the benefit of it's a very similar concept that you're using sequencer to do spatial, but the benefits that they're claiming is for Avidity, they're using their Avidite chemistry, which makes them more efficient. For Singular, they claim the throughput is, or cost is the lowest for all the spatial or all the sequencer market at the time when they're announcing it. So that was the benefit that they're putting into. For both of the technologies, the data set's actually not really publicly available yet. They're just being announced and getting piloted right now. But it's another exciting advancement that you're using sequencers to not just do the sequencing, but on the spatial, using their same flow cell. And one really weird arm on the transcriptomics or sequencing based method side is DiviSeq, which is developed and spun up from Yale at Ron Fant Lab. It's now an Atlas Genomics and the only technology that you can do with if you are interested in doing spatial epigenomics. The concept is you're very similar. You're still doing hybridization captures, but you're attaching microfluidics device on top of the tissue that's already attached to barcode them on like x-axis and y-axis twice, and then collect everything and doing the sequencing. And the way they developed is basically running spatial ATAC-seq and spatial cut and run. The resolution is not high because you're using these kind of like microfluidic devices to kind of force barcode them. But for ATAC or epigenomics applications, you're more interested in nuclear regions, and they claim it's not as important, but this is the only spatial epigenomics products in the market if you're very interested in like how some modifications happen or what are the epigenomic regulators for some of your biological questions. And lastly, going back to LCM technologies, these are some of the examples that are now a little outdated, but still used quite a bit in the clinic. LCM technologies is more of a base of geomics from Bruker, where you can use the protein to stain and image them and select the regions of interest, and using UV to cleave the regions and do the sequencing to do the spatial omics as a classic laser dissection technology. There's a similar concept which is called light-seq, where you're also selecting based on the regions, and you're using lights to basically assign the regions where you're barcoding and collecting after and dissecting the spatial information later. And what's going more of an outlook for spatial sequencing-based methods is more of a temporal measurements. I think sequencing-based technologies has a huge benefit in that because you're going for more targeted. You can do a lot of like screenings or more of a fuller, build a fuller picture on these specific measurements that we're doing with spatial omics. There are a few different ways that people have been doing like more about different time points or different like high throughput screening for using the spatial omics methods. One of the exciting publication recently or a couple years ago was Perturb Map, where you can do spatial proteomics or spatial transcriptomics on the CRISPR library with the barcodes that are already having specific barcodes on the clones, and you can visualize these different clones. Basically, the variety of the paper was you can identify clonal expansion of different tumors within the tissue. Which is a very neat application of spatial transcriptomics technology. With that, it's like 100 different platforms. Any questions, anything confused you or want to know more about? Okay.
Actually, one of the last ones mentioned that's using, is it light to segment?
Yep.
Do you happen to know what like the cellular resolution is?
It's
So
still
it's not one cell.
yeah, it's still more of a micron level. The UV lasers, they claim you can go like a few nanometers level, like 500 nanometer level, but at the end of the day, nobody segments a single cell to because and what we ran on genomics, for example, is they required at least 100 cells to be segmented and do more of a low input sequencing on specific regions. So that's a level of resolution we'll get. But the benefit is you can specifically choose the region.
Shoot lines or
Yep, exactly. A lot of the platforms have its own characteristics on in terms of like throughput, capture area. Well, I didn't really talk about the capture area. Every platform has its own capture area. So sometimes you're very limited to small regions because it's gonna take forever to scan. Or you can take the whole imaging spot with a slightly lower resolution. So there's always like these questions around like how deep you want to go or how large you want to go. But also, as Hannah was asking, like, do you have a custom panel in mind that you really want specific SNPs or specific genes to be covered, or how much you want to how high of a plex you want to go, or whether you want to do multiple RNA and protein at the same time, or different modalities and before or after spatial omics. And of course, with the sequencing, but also for imaging-based technologies as well, having a like fiducial controls, sensitivity, specificity has been a huge part of the debate of like, especially when the first phase of the spatial omics, when each of the companies are saying we're better than the other. But also at the end of the day, it's a question of like how deep or how many reliable probes that you want to get out of these platforms is going to be a question that you might want to ask. So lastly, I'm going to touch upon a little bit on like what kind of considerations you want to have with data processing, and also like end the lecture with one of the example projects that we have in the lab and how we're using spatial ourselves. The spatial omics analysis basically becomes very similar to single cell, where once you have an image acquisition, you are going to use spatial barcodes or spatial images that you had for segmentation or for the expression quantifications back into in a form of a count matrix, where you have a count matrix and you have spatial coordinates attached to it, which becomes very similar to single cell. So you can do expression level analysis, you can do cell typing and GSA or any kind of analysis that you're used to do. But now we can do also tissue specific microenvironment characterizations. You can run like neighborhood analysis, more insight, like spatial variation, like spatially variable genes, or like spatial gradient of specific expression or pathway patterns, which your homework will cover a little bit of that. But now we are able to unlock another round of analysis to ask these spatial specific questions. One common questions that we get a lot, especially from clinical folks, are cell segmentations. Yes, we get a really good quality data sets, we can visualize them, and they always ask like how accurate are these segmentation masks? Because the moment they hear like we're using machine learning algorithm to define the boundaries and map all the quantifications that we have back up to these segmented masks, they're asking like what are the percentages, right? Easy answers depends. Neurons have a really hard time defining the ends of the axons. Crips also have a really hard time defining the ends of the crips, like top of the crips. But generally speaking, if you're dealing with like fibroblasts or dealing with like other epithelial cell types, I would say it's 80% or even like sometimes 90% accurate when it comes to really well-trained model. But sometimes you don't really need a cell segmentation in the first place, like a very early phase spatial transcriptomics methods were claiming. If you were to ask me, I think these are some of the reasons why we care about cell segmentation. like we still want cell segmentation so that we can do single cell level analysis, right? We cannot do interaction analysis if we don't know where the boundaries of the cells are. Like we cannot really understand how, like in a given cell, what kind of cell types are touching and not touching if we don't really have a segmentation test. But also this is more of like a biologically very, very relevant questions that we have to ask. So I always go more for like if we can make a better segmentation, I always tend to do better segmentation if possible and go a little bit of a troubleshooting spot. But I'm also understanding of there are cases that segmentation doesn't matter, especially if you want to see how microenvironments or structures are defined, as I towards the beginning of the lecture talked about histological, cellular, molecular. You're more interested in structures and expression levels of these structures. It might not be as relevant to you. And with that, the challenges include like it sounds easy to segment them. Like when you look at these images from like CellPose or Stardust or like other segmentation algorithm websites, it looks like 100% accurate. What it actually happens if you zoom all the way into these specific segmented masks, you will see like one or two genes that are like getting spilled over from another cells because every cell has a different shape, and also some of the cells have a real, even if you do stain for membranes or if you do stain for like different segmentation 5-6 segmentation markers, sometimes the staining varies and the shapes varies depending the segmentation markers might not be perfect. And because of these, even if we can do multiple exposure to make sure we get the right boundaries, there's still a lot of like errors and room for misassignment for a few of the markers, which can be normally it's fine, but when it comes because B cells tend to stay with B cells together and form a specific structures, right? But when it comes to like tumor normal interface, can be quite troublesome. And when it comes to like germinal centers or follicles, you can see very densely packed regions where the boundaries are not too accurate. You can see like B cells and T cells, for example, like expressing very similar markers. It's probably because the segmentation has not been done perfectly. So there are a little bit of a limitation still, really depending on what cell types you're interested in. And that's the reason behind some of the segmentation-free methods that came out. Bezier and Fixture is one of the major examples of segmentation-free analysis methods. Basically, both of the methods use slightly different statistical idea, but the verdict is they're using pixel level segmentations. So rather than defining each of the cells or each of the gradients and trying to draw the boundaries, they're taking a pixel and cluster this level of pixels with around surrounding information. So and doing the clustering to identify the regions or the cell types or the location type of within a given tissue. And surprisingly, after like a lot of people optimizing, surprisingly it works quite well. You can see different levels of binning and going all the way down to like a single pixel, you can still identify really good structural information. Same with the Bezier, but you won't be able to draw the boundaries and do the cell interaction analysis or understand like what are the interactions that are happening like in this specific regions versus like this specific regions.
Yes. Yep. Some Fixture, but these are visualization methods.
This is more about annotation methods.
Annotation methods.
Yeah.
Okay.
So instead of annotating cell types, you're annotating each pixels. Yeah. So every step even after segmentation, even after you make it into form of a struct object or ScanPy and data objects, you still have to question a lot on like how to really deal with the data sets because sometimes, I mean, when you do sequencing based method, you're dealing with the same idea of negative binomial distribution. But when it comes to probe-based methods, sometimes you're seeing completely different distribution depending on what type of a probe that you're using. So a lot of the cases when you do quality control, I'm giving Cosmics, which is a fish-based protocol as an example. the protein we tend to look into negative probes and look at high and low expression as if you're dealing with antibodies, even if it becomes more of a, even if it takes the probes or it takes a probes to bind it instead of antibodies. For RNA, we tend to use negative and false positive probes as well, but also have the difference is more of like the cell removal part. The min counts per cell and ratio of negative to targets counts is somewhat very similar to single nuclei or single cell sequencing. We still look at the complexity of counts per unique genes, which tends to be a slightly lower than single cell. But more importantly, we do remove the like large or small cells or cells that doesn't contain any information, which is slightly different from single cell because we're taking the size into effect. We also tend to look into specific regions of the tissue for a lot of spatial technologies. We always tend to do spatial projection of the image of the cells, so that we can remove if you're seeing irregularities or if you're seeing specific regions or edges of the tissues to be compromised. We tend to remove them as well. And for probe-based technologies, we tend to also look into the target QC, which nowadays people don't do it anymore. Now that like there has been a lot of data and probes has been optimized enough. But if you're dealing with a new platform or if you're dealing with your own custom panels, I really recommend going through a target QC of like how well your probes are behaving or how similar to these probes are expressed relative to other probe sets that you have in your sample. And then there's almost tons of like spatial analysis, and I think it's still a region field that has been actively developing. There is now different platforms or containers that you can now really start analyzing the spatial omics data sets in the first place, I think, and then in Surat. Like Surat in the beginning, it was really challenging to work with spatial omics data sets because they didn't really have proper image slots. Or else if you start adding images, the Surat objects became too big and hard to operate. Now I think Surat, as well as NData, has been figuring out ways to properly contain like polygon information, spatial coordinates, and all of these informations. But these are some of the examples of what you can do with spatial omics, and this is one of the limited examples that you can use because everyone does spatial omics for their own reason, and a lot of the times they come up with their own analysis methods to do it. But really commonly, a lot of people now, in a standard way, we do a lot of the spatial pattern discovery analysis, right? We look at how spatially varied these given gene or this given gene in a specific cell type look like, do a differential analysis on that variation. There are a few different methods to look into spatial or correlation or variation, which will be covered in homework. But also, I think people are now starting to apply like RNA velocity concept, even if it's not sequencing based. You can look into more of a kinetics by expression levels and variations, but also trying to integrate different methods or different modalities, and coming up like you will see every month you will see like a plethora of different methods that are coming out. Half of them don't work right now, but I think we're going in the right direction of like now starting to use the spatial omics more than us just putting in a last figure and saying like, hey, we have this full imaging. And I think this is also tool development is also aided by a lot of large scale data sets for reference, but also for your biological studies. H10 and HopMap was two big consortia. H10 is more focused on tumor and they're more focused on like immunofluorescent imaging. So they used to have their website still has it. If you look into, they had like an image of the week and look at very cool like immunofluorescent images on their website, and they're starting to put together. They're starting to go more on the RNA side as well. HopMap is also one example, which started like right after COVID or during COVID. That are really going for spatial mapping of the human organ system. A lot of them are started from single cell data sets, but now having expanded to like a lot of the Visium or more of a spatial data sets as well. So it's slowly growing, and their database has been also really helpful. There is Human Cell Atlas recently started collecting spatial data sets as well. Mosaic, which is more of an Auckland initiative, they're trying to also do a similar tumor spatial omics atlasing on few different tumor types to begin with and expanding. There are also like different consortia that are budding, like Gestalt and Stop, which I think Chris and I are part of, that are really aiming to build a community of scientists to develop tools together, troubleshoot, and have these like large data sets in place for to basically for us to use it faster and use the maximize what we can do with these data sets. With that, Cornell also has an atlas project called Saha, which some of you might have heard of it or not. We're currently more of an like we're currently preparing for publication, and that's why if you haven't heard of it, that's probably why. But you will in a few months. And I'll probably end this lecture by introducing the briefly of the data sets, which because I think it's going to be a good demonstration of what you can do with the spatial omics data sets. The real rationale behind this whole atlasing projects are we're still lacking a lot of like data collection, generation of a reference map, especially for spatial. And we have like, as I mentioned, we need for the new methods and tools to be available, we need a really good data sets to understand the statistics behind or understand the way that what's the best way to do this. And of course, we're also very interested in computationally modeling different systems in general, which requires a lot of data sets. And that's why we've been doing spatial atlas of human anatomy projects, which is very audacious efforts to build a reference data from healthy adults with wide range of age, gender, and ancestry across 30 different organs and running spatial omics to understand like these typical spatial omics analysis results that we can get from these samples. We're specifically using Cosmics because that was the highest resolution with highest plex available at the time when we were pursuing this project. So this is one example to see it. Like for Cosmics SMI for RNA and protein, we typically start with H&E and go to pathologists to choose a field of view. We now shine lights on these field of view and have the morphology markers aimed for segmentation. So we typically use 4 to 5 markers for segmentation for Cosmics. And these markers will then help us to segment the tissues and look at the boundaries of the cells like this. And at the same time, we have RNA or protein transcripts or probes bound to these tissue regions, and you can quantify these individual dots or individual binding events and map it back to the segmentation masks. And once it becomes more like a count matrix, it becomes more similar to single cell data sets, but with spatial coordinates so that we can map back and forth. And of course, we can annotate them using single cell reference data sets. There is also in situ type methods from Cosmics that specifically accounts for statistical, like the backgrounds and the background and false positive probes into account. But we also go back to pathologists and experts in the field to make sure our annotations are proper. But then true beauty of the spatial omics dataset is we can go back and forth like UMAP that we can do unsupervised clustering. For example, this is one of the prostate datasets and go all the way to the more of a XY coordinate datasets and go back and forth to make sure we're making sense in both expression space and physical space as well. So this has been like we started as like few investigator at Cornell. It become more of a full-blown project with a bunch of collaborators at Bruker, which is a Cosmics platform provider, NVIDIA, or like other institutions like St. Jude University of Adelaide near Genome Center, where we are kind of all working together to build like, hey, what's the best way to standardize all of these different organs or different workflows for all of these different tissues, but also to what kind of analysis that we can do and how do we really deal with the data sets in the future or databases in the future. So it's been a couple of years since we developed this project. It has become 2 years into the project. We have more than 10 million cells across 100 patients and 20 billion transcripts. And after speaking, we keep generating more data sets. And these are one of the like first 10 organs that we have been focused on, along with some of the organs have disease sets as well to really demonstrate the power of having a good reference data sets. And this is the first snapshot of data sets that we're preparing on publication for. Across these like 9 different organs, we also have PBMCs, which are more of a single cell format. We can go all the way from like H&E structures to a more of a microanatomy structures, and you can basically color each of these H&E structures with the color of the cells or color of niches, and go all the way down to like single cell level, and going back and forth as I mentioned from UMAP to the actual cell types. And each of the organs have like more than 1 million cells each, so that you can use it true as an atlas level data sets. So also the beauty of this data set is it's all generated from the same institutions, from the same workflow, and standardized as much as possible. We can start to integrate the data set and start to question, hey, what are the differences between the B cells in specific organs to the other? So we're kind of, the goal that we're going for is to go from this textbook images, and which you can see somewhat in the H&E images over here, to actual colors of the cell types and neighborhoods that we can detect from these specific tissue types, organs, or structures. And these are some of the examples what you can do with this data sets. This is one example of like Saha appendix data sets, which you have pairs, patches, or like appendix, the colonic patch equivalent lymphoid structures in the regions, and stromal or muscle layers in the back, and crypts with a little bit of a stem cells. You can build a cell type networks of how they are represented in space, or how they're represented in a specific field of view, and so on. But also to see how these networks are not just like how the cells are close together, but what are the means of communication between specific regions that we're seeing over here. And every time we do these analyses, it becomes like huge data sets. And during the process, we also had a lot of questions on standardization, and came up with a spatial touchstone project where we're trying to do multi-center, multi-organ, multi-platform benchmarking study, where we're taking a single block and taking consecutive sections across multiple institutional replicates or platform replicates, and see what are the variations that we're seeing across these replicates that we're generating. And a lot of the findings are still are more of a comparisons of like how we can integrate these RNA and protein data sets, like what are the differences between like Xenium and Cosmics, which is basically ISS and ISH methods. And this is currently in revision at Nature Biotech, so you guys will be able to see it in like a couple months. If you're interested in, let me know. We should be able to share you like a very early version of it. But also the tools that are available as of today is like Spatial QC, where you can automate the metrics that we define in a paper of like, these are some of the metrics that you need to consider when you evaluate the quality of your spatial omics data sets. But also, if you're not familiar with bioinformatics, you can go to like our app and put in your account matrix and calculate it. But also look into what other data sets that are published, and also touchstone data sets and where your data spends, which obviously really varies by organ. And we not only standardize them, we also go for like RNA and protein comparisons, and go for a multimodal data analysis, and start questioning like what we can do with these RNA and protein layers. One example is we take a RNA level cell-cell interaction analysis and validate in a protein layer. They're seeing region-specific interaction patterns and concordance with the protein data sets, as you can see over here. But also start to merge them and help them to further annotate cell types. This is one example where we ran H&E, RNA, and protein on the same exact slide, doing a true multi-omics on RNA and protein, and merge the data sets together so that we can not only look at the RNA level annotations that we often do with single cell, we can cluster them and annotate them, but adding the protein layer actually helps us to further define similar cells into a like subtypes by looking at their phenotypes. And not just we're doing like multiple modalities or multiple benchmarkings, we also are very interested in generating more of AI models to train these large data sets to help people use this or utilize it to predict, annotate your own spatial data sets, but integrate your own spatial omics data sets with the batch integrations. There are a few different ways to do it, and we're currently collaborating with NVIDIA to train our datasets so that people can get better access to without having to generate their own data store, without having to go through hard, complicated computational analysis if you're not familiar with spatial omics. But we're not the only one doing it. I think there are a few companies and tools that are out there that are trying to do like virtual identity staining or train different image models to look at like annotations easier. So I'm sure that within a year or two, there's going to be a ton of different foundation models that can be used for your own data sets as well. We are also planning on generating, increasing the accessibility, not just by generating models, but to have visualizer. This is one example of what we did for astronaut data sets on like different single cell metabolomics, proteomics, and exosome stuff. We're building the same thing for Saha right now so that people can go in and look at the images, select the regions that they're interested in, look at the expression levels, and play around with the data and start asking questions depending on what you're interested in or what cell type you're interested in. And I'll end the lecture by really showing how it's actually being applied in a real research setting, not just like having a lot of data or having big database. Like one example is you're looking at colorectal cancer samples, for example. You can have very similar regions of tissues that are like they're all crypts. When you look at spatial omics, you're seeing quite different crypts, or you can see almost like how deep the crypt cuts look like if you're trying to do it with spatial transcriptomics. But also all the tumors that we're detecting over here, we can now see how the lymphoid tumor ratio varies across within the regions, although there's a tumor regions that you can all collect, or you can do bulk RNA sequencing or single cell on. You can now see these specific tumors that are arising at like high lymphocyte to tumor ratio, regions versus not. And you can start to identify like really novel features that you haven't been able to see before. This is one example one of our grad students found, where you're seeing the nerve regions and the tumor cells that are surrounding over it, and which is nicely shown in the spatial omics data sets. And if you were to run single cell, this level of number of cells will not be detected in single cell in the first place, but also even if it's detected, it's going to be like another glial cells because we don't know where it's actually coming from. And you can do interesting analysis of like how these tumor cells are interacting with these specific nerve cells, and so on. And even at the clinical setting, you can look into like this is one of the studies that we did in collaboration with another lab looking at early stage PDAC. Basically, every lab, like a lot of your studies look like, hey, we have this interesting disease that we want to study. You can do like bulk RNA sequencing on a large cohort. You can do tumor organoids. But now we're starting to add spatial profiling as well. That you can still do like expression pattern analysis as you were to do in bulk RNA sequencing. But you can now project some of these information onto spatial coordinates. You can see specific T cell environments tend to like farther spread apart in specific genotype over the other, and start to question, okay, what are the unique immune microenvironment that differentiate different subtypes of the tumor? And I mean, we're not the only one who's doing it. A lot of if you look at every tumor atlas, if you look at a lot of research studies, people are now starting to look into like spatial colocalizations in addition to bulk RNA sequencing or interact analysis that they're doing. And I believe that this will eventually, I think, everyone's really looking into how we can apply this in a form of therapy decisions or really finding a good target for immunotherapies or other treatment options for the patients. With that, I think this is the way we're going forward as like spatial researchers, but also what we are personally interested in, is like we're still finding different patterns within tumors, within the normal organs. And now we're also at like a lot of the conversations are now moved from single cell to niche and microenvironment, like tumor microenvironment dynamic has been quite a bit of hot field in the past few years or even more. And now we're starting to look into how we can connect these microenvironments into like tissue networks and organs. I think eventually the Saha project, I was really passionate about the Saha project as we can do start to add and connect different systems together and start asking questions around tumor metastasis, immune organ networks, and systemic diseases. These are something that would be possible as we start to do more, but unbiased, like spatial omics profiling. Yeah. And as we're developing different databases and generating more data sets, we're still at like data integration aspects. And in terms of atlasing data sets or generating more data, we are also partially working on scalability and cost of this thing on spatial omics. So in the beginning of the lecture, I asked you guys, like, why would you want to do spatial omics? And a lot of the cases, the questions were very much around, like, hey, I'm interested in the specific structures, which was the case for, like, when I asked my collaborators and they came up to me and I'm like, why do you need spatial in first? Like, why don't you do single cell? And now we're shifting our arguments a little bit and start also doing spatial on something that we would normally do single cell because of the scalability of the cost. Which based on like a lot of the new technologies like STEM, which basically is the same concept as spatial omics, but we're stamping the cells onto the slide before we do the spatial, before we do any kind of like preps and do the spatial. And what this method or approach gives us is we're getting higher resolution than what we would do in a single cell, right? We can image the cells and look at the individual structures and sizes and areas and morphologies of the cells and quantify them and have the exact same single cell level information. But also because we're loading so many cells, whereas like if you were to do single cell, you do maximum of 10,000 cells per library, you're doing millions of cells in this slide, and you're now able to understand and identify really rare cell types. For example, this is an example of like circulating tumor cells. You can start to identify really rare cell types that you cannot identify by simply running single cell. And as we're going for all the way to full transcriptome, which you're seeing a huge, really good correlation with single cell RNA sequencing, we are now developing methods to like, hey, if you want to do PDMC instead of PDMC single cell, we're running PDMC spatial. This was actually done on astronaut samples. You probably know Chris does a lot of like astronaut sample studies. These are our optimization on really like we're putting for each samples, we're putting more than 250 cells per stent, and this became one sample per stent or substent. And in total, for each of the whole slides, we're getting around 3 million cells, or like around this number to, which is a far more number that we're getting. If you do the cost calculations, we're actually talking about really like fraction of the cost of the single cell RNA sequencing to spatial, even if you're considering we're doing whole transcriptome. And we're getting additional information on segmentation markers and how the cells look. So this is one of the examples which I guess I'm running out of time, where we were trying to do single cell, instead of single cell, we're doing spatial. And although the numbers are like you're getting a slightly lower complexity of the data sets, you're getting 40 times more number than if you were to do single cell on the same exact study. And one additional benefit over here is this is like a sample that we collected during the flight when they're actually on space flights. And these samples that were collected over different tubes probably would have survived the single cell library prep, whereas we just fix them away and run spatial. We were able to collect these amount of samples already to use for our biological information. We're seeing consistent performance across different replicates and samples. And when we do the annotations with conventional single cell methods, we're getting very similar annotation level to like any reference PBMC datasets, with a lot of the cells that are intermediary as well, but also a lot of the cells that like pluripotent stem cells as well as plasmablasts, which we oftentimes don't really capture a lot in a regular single cell data sets. So skipping apart, there is like tons of different ways that we're using spatial into a new way to scale things up, but also ask new questions. And we get to revisit different structures that we haven't been seeing in the H&E thanks to spatial. Like we're starting to really correlate these aging structures in H&E into like RNA level because we have both RNA and H&E images. But we are also building different network structures that start asking questions like never before, but also all the way going into the subcellular, how the proteins correlate, co-localize within the cell, and going all the way down to subcellular level information, which here we're finding very specific co-localization pattern only during flight in our space-blown PBXEs. And we're eventually really going for like building similar graphs and maps for human as well. This is one example of like every biological process in the East. And I have been telling people like we can soon do it at human as well. Not in a form like in a single paper like this, but in a form of like a huge model. But I think we're getting there. That these are some of the acknowledgements about all the people who've been part of the projects. With that, yeah, that's the end of the lecture. Hopefully this was a little bit of a demonstration of what we can do and ideas that we can have. But if you guys have any questions, feel free to ask.